Welcome to the next episode of the Microbiology Tube. So today we'll be discussing about the different biochemical tests that is used in the microbiological laboratory. So I'm just going through the quick review on this biochemical test. So I don't want to discuss about the detail because you need is just for your practical viva. So first I will discuss you about the indoor test. So indoor test is just type of test in which the tryptophan that is used in the media are converted into the indole. So if the bacteria is able to produce the tryptophanase enzymes, then this will convert the tryptophan into the indole. So some of the bacteria are there that can synthesize or that can convert the bacteria that can synthesize the tryptophanase. So that bacteria will convert the tryptophan into the indole. But if the bacteria are unable to produce the tryptophanase enzymes, then it won't convert the tryptophan into the indole. So this is the basis. So what happens is that in the indole, if we add the Kovacs reagent, so if we add the Kovacs reagents, so that it will give the cherry color ring compound in the test tube. So if this is the test tube, so in the surface of the test tube there will be the cherry red ring formation. So if there is the cherry red ring formation, so this is called the indole positive bacteria. So the indole positive bacteria are E. coli, Proteus, vulgaris, Proteus, Morganella morgani, Providencia species, etc. So we can also differentiate between the species by use of the indole test. For example, if there is a confusion between whether the bacteria is Klebsiella oxytoca or pneumoniae, so if we do the indole test, so then after that can be differentiated from the Klebsiella oxytoca and the Klebsiella pneumoniae. So Klebsiella oxytoca are indole positive, Klebsiella pneumoniae are the indole negative. Cytobacter cocci are the indole positive, whereas the Cytobacter frundi are indole negative. So Proteus vulgaris are the indole positive, whereas the Proteus mirabilis is the indole negative. So one of the very simple techniques to remember is the OKVIP. So O means the oxytoca, which means the Klebsiella oxytoca, K means the cocci, which means Cytobacter cocci, and V means vulgari, that means the Proteus vulgaris, and this IP last stands for the indole positive. So if you can remember this formula, then you can remember this all things. So next is the methyl red test. In the methyl red test, what we do is that we look whether the glucose is converted into the acid or not. So some of the bacteria are there. So what does this bacteria will do? That bacteria will convert the glucose into the acid. So what we do, we have the media called the MRBP medium. So in the one test in the MRBP medium, we will put the bacteria, and after the incubations at 18 hours at 37 degrees centigrade, we will add the indicator called the methyl red. So if there is the formations of the acid, then what happens is that if there is the formations of the acid, so the methyl red indicator will change. So methyl red indicator will change from its yellow color to the red color. So if there is the low pH, then what happens is that in the acidic conditions, the methyl red will change its color from yellow to the red color. So next, the UMR positive bacteria are the Escherichia coli, Salmonella, and the Proteus species. So next is the Bogus postcard test. In the Bogus postcard test, we will be detecting whether the bacteria is able to produce the acetyl methyl carbinol or not. So what happens is that the glucose will be converted into the acetyl methyl carbinol. Some of the bacteria are there which are responsible for converting the glucose into the acetyl methyl carbinol or not. So if there is the conversion from the glucose to the acetyl methyl vinyl carbonyl, then in the presence of the alpha naphthalone, so in the presence of the alpha naphthal, what happens is that this acetyl will be converted into the diacetyl. So diations. So what happens is that if you add the acetyl methyl carbinol, so if there is the acetyl methyl carbinol and if you put the alpha naphthalone, it will be converted into the red coloration. So that is the basis of the Bogus postcard test. So in the methyl red test, the glucose was converted into the acidic conditions into the acid, whereas in the MRBP test, the glucose is converted into the acetyl methyl carbinol, which is called the acetone. So this is the negative VP test and this is the positive VP test. So the positive, the bacteria which gives the BV positive are the Listeria, Enterobacter, Viridans group, Streptococci, Klebsiella, Serratia, Apnea, Vibrio altor, Vibrio agnolyticus. So these are the bacteria which are Bogus postcard positive test. So citrate test. So next is the citrate test. So in this test we will detect whether the bacteria is able to produce the citrate permease enzymes or not. If the bacteria is able to produce the citrate permease enzymes, so in the medium there will be only the citrate. So the citrate will be the food for the only food for the bacteria, and if the bacteria is able to produce the citrate permease, then the citrate will be changed into the pyruvate, and this is called the energy source. And if the citrate is converted into the pyruvate, then the bacteria will metabolize and it will convert the ammonia salt into the ammonia. So if there is the more amount of the ammonia, it means the pH of the medium has increased. So if the pH of the medium has increased, then what happens? We are using the bromothiamyl blue as the indicator. So the bromothiamyl blue indicator will change its color from green to blue. So if the pH is above 4.5, above 7.6. So this is the negative one. So initial color of the citrate, and this is the positive one, the final color of the final color for the positive bacteria. So next is the urease test. So what happens is that in the urease test, so some of the bacteria are there that is capable of synthesizing the urease enzymes. If the bacteria is capable of synthesizing the urease enzymes, it will convert the urea into the ammonia. So if the urea is converted into the ammonia, just like in the citrate test, the alkalinity will be increased. It means the pH will be increased. So if the pH will be increased, then what happens? We are using the phenol red as the indicator here. So phenol red indicator will change its color from 6.8 to from orange to the pink color. So at the pH 6.8, below the 6.8 it was orange color, but whenever the pH rises above 8.1, then what happens is that the phenol red will convert its color from orange to the pink color. So the urease positive bacteria are the Proteus, Corynebacterium, Helicobacter, Yersinia, and the Brucella, and the Cryptococcus are the yeast. This belongs to the fungi. So the Cryptococcus are the first urease positive organisms. So these are the Cryptococcus is the yeast of the bacteria. So thank you for watching my video. This is the very quick review of the biochemical test. If you want to get the detail of this biochemical test, I have in my other videos too. So this is only for the practical exam preparations. I hope you like it. So if you really like my video, please don't forget to subscribe the channel and like and share the video. Thank you.